Thank you very much for the organizing committee of the 4th Digital Congress on Nanobiotechnology and Bioengineering for giving me the opportunity to present some of the work that we are doing in our lab. Today I'm going to talk about biogenic synthesis and application of nanomaterials. Biogenic synthesis is the process of synthesizing nanomaterials using biological materials such as plant, microorganisms, or fungi. This process is often referred to as green or sustainable synthesis because it does not use harmful chemicals or solvents. Green chemistry contributes to the sustainability of chemical process, energy saving, lesser toxicity of resin and final products, lesser damage to the environment and human health, decreasing the risk of global overheating, and more rational use of natural resources. Before getting into my talk, I would like to tell you that our lab is in the Faculty of Pharmacy and Biochemistry of the University of Buenos Aires, and most of us are also CONICET researchers. The topic of interest of our team includes the development of biomaterials and nanomaterials for biomedical applications. We are also interested in stimuli-responsive materials, and we also incorporated the 3D printing technology. There are several reasons why biogenic synthesis of nanomaterials is important. First, it is a more environmentally friendly process than traditional chemical synthesis. Second, it can produce nanomaterial with unique properties that are not possible to achieve with traditional methods. Third, it can be used to produce nanomaterials that are biocompatible and can be used in different applications. Over the past 2 decades, the development of green synthesis procedures experienced a great development thanks to the contribution of researchers of diverse origins. The driving force of this development was the possibility to employ environmentally friendly and sustainable processes. These green synthesis procedures require biocompatible reagents, non-toxic solvents, and mild reaction conditions such as low temperature, adequate pH, and controlled ionic strength. Moreover, slight changes in experimental conditions such as precursor concentration, pH, or additives can lead to substantial modification of the resulting nanomaterial. The broad field of green synthesis research is now well established as denoted by several publications, including research articles, reviews, and books. Indeed, the number of publications in the field of green synthesis experienced a tenfold increase during the last decade. At the forefront of research in this area is the development of methods for the synthesis of nanomaterials. Especially during the last 5 years, most of the articles published in the field of green chemistry were associated with nanomaterials. The performance or function of the resulting nanomaterials strongly depends on the size, shape, and surface properties, among other important characteristics. Indeed, a proper characterization of the resulting nanomaterials requires the simultaneous or subsequent application of various analytical methods. Green synthesis procedures can be used to produce complex nanostructures with interesting applications, for example in catalysis or drug delivery. These nanomaterial synthesis procedures have been focused by researchers. Indeed, there is a growing interest in the development of eco-friendly, cost-effective, and direct methods for the synthesis of nanomaterials. Moreover, the versatility of green chemistry allows producing a wide range of nanomaterials with numerous promising applications. Indeed, during the last years, we were working in this field, producing nanomaterials with different components such as iron, zinc, copper, silver, among others. One of our first work in this field was the green synthesis of silver nanoparticles employing yerba mate plant aqueous extract. Yerba mate dried leaves are highly available and inexpensive industrial product in South America and other countries. Yerba mate plant aqueous extract was used as a reducing reactant and stabilizer agent due to its great content of phenolic compounds. Different parameters such as silver salt and reducing agent concentration, pH, temperature, and capping agent concentration were evaluated to establish the optimal synthesis condition. Yerba mate plant extract was used as a reducing and stabilizer agent due to its great content of phenolic compounds. In the first step, silver nanoparticles were obtained through a chemical reduction of silver ions employing the phytochemical present in the aqueous extract. In this way, it was possible to obtain nanoparticles of around 20 nanometers in size. In a second step, we absorbed some of these phytochemicals present in the aqueous extract on the surface of the nanoparticles. This step was very important to stabilize the nanoparticles. The minimum inhibitory concentration for the different microorganisms, including a gram-positive, a gram-negative, and a fungi, was analyzed. It was observed that the nanoparticles possess antibacterial and antifungal activity. This effect was higher against the gram-negative bacteria, as it was reported in several studies. Moreover, the present nanoparticles presented antioxidant activity as several green synthesized nanomaterials, where phytochemicals are absorbed on the surface of the nanoparticles. The resulting nanoparticles possess antimicrobial activity due to the silver component of the particles and the release of silver ions from these particles. The nanoparticles also possess antioxidant effects due to the phytochemicals absorbed on the surface of the nanoparticles. These molecules also contribute to the stabilization of the nanoparticles and also contribute to the improved biocompatibility of the nanoparticles. In conclusion, we developed an economic process to produce potent antimicrobial, antioxidant, and biocompatible nanomaterials. These properties are strongly essential for biomedical applications. In a similar approach, different copper and zinc nanostructures were obtained using yerba mate plant extracts as a reducing and capping agent. Furthermore, adjusting the sodium chloride concentration and temperature, it was possible to successfully tune and examine the morphology of the resulting nanostructures. In this sense, you can see here how different are the structures obtained with each component. For example, with copper, we obtain these highly porous materials, while with zinc, we obtain this kind of nanosheets. Our studies show a higher bactericidal activity of copper-based nanomaterials against gram-negative bacteria than zinc-based nanomaterials. However, in all cases, the nanomaterials were able to reduce nearly 99% of the viable bacteria after 24 hours. In addition, we also observed the antioxidant activity of the nanoparticles, mainly due to the presence of phytocomponents from the plant extract absorbed on the surface of the nanoparticles. In a collaboration work performed with a group of Professor Hotanya from India, we employed different plants to synthesize calcium ferrite powders. The Aciderata indica, commonly known as neem in India, is a tree native of India. Almost of all parts of this tree are used typically for curing fever, inflammation, infection, skin diseases, and dental disorders, due to its antimicrobial, antioxidant, anti-inflammatory, and antifungal properties. On the other hand, Murasha kengi, known as curry, is a plant from India, Sri Lanka, Pakistan, and China. It is also a rich source of alkaloids, carbohydrates, flavonoids, and sterols. The results obtained in the antioxidant and phenolic acid study showed the potential of both plant extracts as a source of natural antioxidants. It is worth mentioning that polyphenols and other phytochemicals present in the plant extract can work as surfactants, reducing and stabilizing agents. Scanning electron microscopy of both samples showed the spongy-like structure. In addition, the electric measurements showed the frequency-dependent behavior of both samples. And the ferrites prepared in the presence of both extracts show magnetically soft nature with multi-domain structure. The saturation magnetization of the powder prepared with neem leaf extract was observed to be higher than the value obtained for the sample prepared with the curry plant extract. The aqueous extract of Aciderata indica is characterized by the presence of saponins having the highest concentration, while tannins and glycosides are present in moderate concentration and low concentration of alkaloids, flavonoids, and reducing sugars. Alternatively, the phytochemical analysis of Murasha kengi revealed the aqueous extract of leaves contains high quantities of flavonoids, alkaloids, and tannins, with low levels of saponin and carotenoids. The molecules of saponin possess a carbohydrate portion, making them water-soluble. Due to this, they have surface-active and detergent properties. Finally, tannins possess numerous functional groups, mainly hydroxyl groups, with the ability to bind different compounds. In this work, it was confirmed that the difference in the phytochemical form in both plant extracts played a vital role in deciding the resulting properties of the green synthesized calcium hexaferrite. I would like to mention also this second work performed with a group of Professor Hotania from India, in which we prepare M-type hexaferrite and spinel nanoferrite powders employing plant flower extracts. This plant possesses phytochemicals that can work also as caping agents to enhance the dispersion and reduce the agglomeration of nanoparticles. In this way, the average crystalline size of the nanocomposites obtained was between 25 and 44 nanometers. Concerning the application of green synthesized nanoparticles, I would like to mention this work performed in collaboration with a group of Professor Ratiram Chowdhury from India, in which bismuth nanoparticles had been synthesized using a flower plant extract. These nanoparticles possess a spherical shape with a diameter between 10 and 20 nanometers and a surface area of 71 square meters per gram. These nanoparticles were employed for the degradation of two toxic dyes. One was thymol blue and the other Congo red, under visible light irradiation. The study showed that 98% degradation of thymol blue in 40 minutes and 70% degradation of Congo red in 80 minutes. Moreover, it was possible to reuse these nanoparticles for the degradation of the dyes under several cycles without losing their photocatalytic activity. In recent years, nanomaterials, with a focus on their biomedical and nanotoxicological effects, have been a topic of great development and serious concern. The synthesis of nanoparticles is generally carried out under controlled thermodynamic conditions and from atoms or molecules. In this way, nanoparticles specially designed for various applications could be obtained. In all cases, the physical and chemical properties are extremely important since they will determine the correct function of the nanoparticles or the products that contain them, as well as their degree of toxicity or safety. On the other hand, there is a growing interest in the toxicological effects of nanomaterials because they can cause toxicological effects through mechanisms not described so far, ranging from the simple physical union to cells to the triggering of complex processes that led to an elevation of oxidative stress. In this way, we can be exposed to nanoparticles during the production, the use, or the disposal of the products that contain nanoparticles. Indeed, we change from a situation where we were rarely exposed to nanoparticles to a situation that we are usually exposed to products that contain nanoparticles. And in this case, different properties and characteristics of the nanoparticles, like the size, shape, structure, can determine their function or their effects. They can reach the environment where the different environmental factors could affect their behavior, and finally they can impact on the human and biota through different mechanisms, through oxidative stress, to bioaccumulation or genotoxicity. Nanoparticles are widely used for biomedical purposes, but also in different products such as food, paints, cosmetics, and industries. Considering their mega production, we should not ignore their potential hazardous effects on human flora and fauna. Indeed, the toxic effects of nanoparticles on aquatic organisms were reported, differentiated in algae, crustaceans, mussels, fish, and others. Cells have been proposed as a unique target group in nanotoxicology, both for being abundant in coastal marine and estuarine environments, and for their feeding suspension behavior, which in turn implicit their ability to filter large amounts of water. Such features enable the retention of both chemicals and particles, including those at the nanoscale. In this case, we observed the dose-dependent accumulation of silver nanoparticles in soft tissues of mussels. In this fish model, we also observe a time-dependent accumulation of silver in the gills with oxidative stress and detrimental physiological effects. We also observe the accumulation of silver in other tissues like brain, liver, and gills, where the activity of various enzymes involved in the antioxidant defense were significantly affected. The mechanism of toxicity of nanoparticles is mainly due to the strong affinity of silver ions for sulfhydryl amine carbohydrate groups, although competition with enzyme cofactors may also contribute. These mechanisms involve the naturing of proteins and enzymes, leading to cellular disruption and structural abnormalities. In the case of biogenic synthesis of nanoparticles, we also evaluated the toxicity of these nanoparticles towards different types of cells, and we also observed that there is a toxicological effects of these nanoparticles over different microorganisms. This work was also performed with the group of Professor Ratiram from India, and it was interesting to note that also biogenic nanoparticles present a dose-dependent effect. However, in most cases, if the green synthesis keeps the molecules employed during the synthesis that contribute to stabilize the particles, the mechanisms and the toxicity of the nanoparticles could be lower. In this sense, It is worth to mention that during the production of these nanoparticles, the material was subjected to a calcination step where all the organic molecules were removed from the nanoparticles, thus they are no longer present in the surface of the particles. Indeed, superoxide dismutase and peroxidase activities were significantly affected by the presence of these nanoparticles. Indeed, the activity of both enzymes increases with the increase in the amount of the nanoparticles employed. In the same way, the viability of the cell was significantly affected by the presence of the nanoparticles. Indeed, after 4 hours incubation with the nanoparticles, the number of viable cells was significantly lower when compared to the control cells. In recent work, we highlight the decrease in the release of silver ions observed when the humic acids were present in the environmental media. The decrease in accumulation and toxicity of silver nanoparticles could be due to the stabilization and complexation of the silver with the humic acid compounds. Thus, they mitigate the toxicity from silver nanoparticles. In conclusion, the biogenic synthesis of nanoparticles or the green synthesis of nanoparticles represents a land of complex nanostructure when it's possible to obtain nanostructure or nanoparticles employing different materials and different synthesis conditions. In all cases, it is really important to make a clear characterization of the nanoparticles employing different analytical methods to obtain a real understanding of their structure and function. Biological sources used in green procedures, for example plant, microorganisms, animal products, introduce a great complexity derived from their structure, composition, and functionality. Indeed, green components can play different key roles in the synthesis of nanostructures, and variations in the synthesis conditions, such as pH, temperature, the concentration of precursors, among others, can lead to different structures with different functionalities. As a result, very attractive, complex, and diverse nanostructures can be generated. So, identifying key biomolecules and conditions is central to the reproducibility of these procedures. It is therefore of paramount importance to emphasize the interplay among the green sources, the synthesis conditions, the resulting nanostructures, and their properties and applications. For our next steps, we want to enhance the process of learning and teaching this technology. Especially, we participate in different undergraduate and postgraduate courses. We want to expand research frontiers and outreach activities in our lab, foster partnership and collaboration with different groups to improve this field of research, and in this way we want to be able to generate an impact on this field. Finally, I want to acknowledge the work of all my team here in Argentina who developed most of this work, our researchers, PhD students, and graduate students, and a special thanks to our collaboration from Argentina and abroad. Thanks for your attention and do not hesitate to get in contact if you have any questions.